Today we'll be studying cholera. What is cholera? To understand cholera, we first need to know what are vibrios. Okay, vibrios are gram-negative bacilli. They are actively motile. The very significant feature of this bacteria is vibratory motility. Okay, which is drawn by what? See, it has this single polar flagellum, which uses it for vibratory motility. Okay. Vibrios were first observed by Robert Koch in 1886, and he also named it as coma bacillus because of its curved shape. They are ubiquitous. They are found in what? Mostly because whatever epidemics that have occurred, whatever pandemics that have occurred, they are because of sewage drawn into rivers and their affinity towards saline water. So, what is the habitat of vibrios? They are found in marine environments, sea waters, seafood, surface waters, rivers and sewages. So, these especially Vibrio cholerae, they cause devastating acute diarrheal disease, that is cholera. And they have been seen to cause 7 global pandemics since 1918s and several epidemics also over the centuries. What is pandemic? What is endemic? Endemics is certain base cases that are always present to the certain region. Epidemic is when these cases rise in numbers more than expected. And pandemic is affecting several regions together, large areas together, right? Vibrios are classified based on their halophilic nature. What is halophilic? Ability to survive in higher salt concentrations. So, those who survive in higher salt concentrations, those are known as halophilic vibrios. Okay. They both require some salt concentration, which is 0.5 to 1% in both the cases. But these halophilic vibrios, they can withstand 7 to 10% of salt concentrations. What are the examples? Parahemolyticus, alginolyticus and vulnificus. Then coming to non-halophilic vibrios, they are Vibrio cholerae and Vibrio mimicus. Based on their antigens and phenotypic and biochemical reactions and serotyping, they are also described as, what is this? O1. O1 is a large serogroup which is divided into this group based on polysaccharide, O polysaccharide, which is present on the bacteria. Okay, it might contain from O1 to O200, but out of these only O1 and O139 were able to cause cholera. Okay, that's how we differentiate them on a larger group known as serogroup, that is O1 serogroup and O139 serogroup. Then there are certain biotypes. Biotypes are differentiated based on what? Their chemical reactions, biochemical reactions, phenotypes, their susceptibility to certain antibiotics. Based on that, we'll differentiate them in classical and LTOR. LTOR is a name of a place where it was first identified. And first 6 pandemics were caused by classical, but in current situation, LTOR is propagating. Okay? And this O1 is further classified into serotypes as Ogawa, Inojima, Hikojima and Inaba. So, Ogawa and Inaba is followed by Ogawa, but if both of these two traits are present in a certain organism, we will differentiate them into Hikojima. Okay? This is the classification based on Gardner and Venkatraman. I told you about the O1 serogroup. It is agglutinated by O1 antisera. How will you perform this reaction? You will take the culture, from culture plate you will take the colonies, you will react these colonies with the O1 antisera, which is available in the laboratory. If there is agglutination seen, it means this particular organism belongs to O1 serogroup. Okay? When there were non-agglutinable vibrios, they were previously classified as non-agglutinable vibrios, but this classification is not in use now. We are only differentiating them based on O1 antisera as O1 and O139. Okay. Out of these, I told you that O139 has been since 1992. This was identified in Kolkata. Then coastal India and Bangladesh, it has been causing several epidemics and outbreaks since 1992. Other than this, if there is non-O1 or O139 serogroup, they have been seen to cause sporadic. Sporadic means rare outbreaks in India. But they do not cause epidemic cholera. Okay, this is because their pathogenicity is low. This is how you differentiate classical and L-TOR Vibrio cholerae based on their characteristic traits. For example, you will see that most of the things are on negative side in classical biotype. It does not show beta hemolysis on sheep blood agar. Tick erythrocyte agglutination does not take place with classical biotype. It is susceptible to polymyxin B. L-TOR is resistant to polymyxin B. Group 4 phage susceptibility. L-TOR phage 5 susceptibility. These are phages that restrict the organism's growth. Then Boak's-Proshicure test. It is the test to see acetoin production in the biochemical. When you inoculate this bacterium into glucose phosphate broth and add certain reactive agents, you will see the pinkish color change in the biochemical. So that is also negative in classical biotype. And cholera toxin gene, in classical biotype you will see CTX1 and CTX2 in L-TOR. Okay, about pathogenesis of cholera. It is toxin mediated. You know that it is, the mode of transmission is ingestion of contaminated food and water. What is the infective dose? It has to be a very high infective dose because this organism is acid labile. It means it gets destroyed in your stomach if it's in the small amount. So more than 10 to power 8 bacilli required to bypass this gastric barrier. Then what will happen? Where does it affect? It's a non-inflammatory diarrhea which affects your small intestine. So there are certain factors that promote your, where your gastric acidity is reduced, such as you're taking antacids, achlorhydria, and what are the pathogenic traits of this organism? It has certain proteolytic enzymes which helps it to get into the enterocytes. So through enzymes and through toxin. What is it? Toxin? Cholera gene. Cholera toxin. Through cholera toxin, it acts on your intestine. See here. What is this thing? These are all enterocytes, right? On these enterocytes, the cholera toxin has come and attached. Cholera toxin has two units. First is A, which is active unit, and B, which is binding unit. So without binding unit, active unit cannot get into the cell. So first thing is this binding unit gets binded to the enterocyte and then active unit through endocytosis enters the cell. After this it goes to the endoplasmic reticulum and from there it goes back into the cytosol. From cytosol, in cytosol what does it do? It acts on the G1 gangliosides. It is acting on what first? G1 ganglioside. I told you about enterocyte. This G1 ganglioside is a receptor that is present on the enterocyte. Okay, don't get confused with the G protein. After that, in cytosol, it reacts on the G protein alpha, which is also a self-regulatory protein which will regulate the cyclic AMP in these cells. So when there is cyclic AMP is activated, this loses its ability to auto-regulate it. Because of active cholera toxin, it ribosylates this G protein. Okay, and then this cyclic AMP remains in the active state. What happens then? All the chloride ions, they are released into the intestine, and to maintain this osmotic pressure, water from these cells will also go out. And this is the reason you usually observe rice water stool, because there is no fecal content, there is only water and ions, right? That was about the mechanism of cholera toxin. There are certain other virulence factors such as zona occludens. They disrupt the tight junction between the enterocytes. Accessory colonization factors that help them in adhesion and colonization into the cells. And LPS we discussed, but this is not used, this is only used for O polysaccharide and LPS, it's only used for diagnosis of the bacteria, and it is also a component in vaccines. Okay. What are the clinical manifestations? In about 75% of the cases, this remains asymptomatic. Okay. But once there is sudden onset of explosive and life-threatening diarrhea, you will see that it's painless. Painless, watery diarrhea. What kind of diarrhea? This is the stool sample which you can see in cholera. What does it look like? It also looks like the rice had been washed in this water, right? When you wash rice, it takes up this color. So it is known as rice water stool. So this thing you have to remember. It has watery, it has mucus flakes and inoffensive odor. Vomiting may be present, but fever is absent. How does the disease progress? When there is about 5% fluid loss from the body, you will notice that there is increased thirst. At about 5 to 10% fluid loss from the body, you will see postural hypertension, weakness, tachycardia, and decreased skin turgor. What is decreased skin turgor? When you try to lift the skin, pinch the skin, it will not come to the normal form quickly. Like it will sustain there, the wrinkles will sustain. That is decreased skin turgor. Okay. When there is more than 10% fluid loss from the water, what will you see? You will see oliguria. What is oliguria? Decreased urine volume. Okay, then you will see that the pulses are weak, feeble pulse, sunken eyes. In fontanelles, you will see, in infants, you will see the fontanelles have sunken. And what is this? Washerwoman skin. Again, wrinkled fingers. Okay. Somnolence is sleepiness, and subsequently, person might land into coma. Okay. Looking at the history of cholera, there have been 6 pandemics since 1817. It originated where? Since 1923 to 1961, it was restricted to homeland. But after this, since 1961, cholera had been found everywhere in the world. The 7 pandemics that have occurred, out of these 6 pandemics were because of the classical biotype, and in 1964, by 1991, the only pandemic that was caused by L-TOR. Everybody knows that cholera is a notifiable disease. What do you mean by notifiable? Notifiable is something, when you find the case, you have to inform the government that there is this case and we have to find the source of infection. Then certain actions will be taken. We'll find out the source from where this cholera outbreak is seen. If it's from the well, they will stop using water from this well and treat the well accordingly. Okay. About 94 lakh deaths have been seen every year because of this worldwide. Majority of cases are due to O1 L-TOR in current situation. We have this National Institute of Cholera and Enteric Diseases in Kolkata, which looks after the cholera in India. Human is the only reservoir. Asymptomatic cases or carriers are the main source of infection. It is mostly seen in high temperatures, heavy rainfall and flooding. Other factors that can cause cholera is poor sanitation, poverty, overcrowding, population mobility, such as there is pilgrim fairs or festivals or even the marriages. What are the certain factors that determine severity of the disease? It is seen people with blood group O are seen to have cholera more than other blood groups. Just remember O1 and blood O group. Lab diagnosis. What samples you will take for this? You will take stool sample and rectal swabs. So if you are not able to transfer these to the laboratory in optimum time, you have to transport them into the transport medium. What are these transport mediums? They are mostly high saline, salt saline containing mediums. So for this, you have to clinically diagnose cholera first. Based on your suspicion, you will send these samples in transport medium. In direct microscopy, you will be able to observe gram-negative rods. They are short-curved, comma-shaped, arranged in the parallel rows. That gives them the name fish in stream appearance. Okay, and there is very important diagnostic facility, that is hanging drop method. In this method, you will be able to observe the bacterium whose direction of travel you will not be able to track under the microscope. So it has this shooting star vibratory motility. Based on that appearance, you will be able to diagnose that this is Vibrio cholerae. If you see gram-negative bacterium but this trait is absent, it means it is not a Vibrio cholerae. This is how they are observed in gram stain, gram-negative. If delay is expected in the stool sample, you will send the stool sample in these transport medium. Don't forget to observe this autoclaved sea water. Even the sea water, which is autoclave, you can use this as a transport medium. It is aerobic or facultatively anaerobic, non-fastidious and grows well on the ordinary medium such as nutrient agar, but in stool samples there are enteric pathogens also, enteric organisms also, which can grow on these plates, and that's why we have to use selective media for classifying this Vibrio from the others. So for that you will use, on McConkey agar you will be able to observe translucent non-lactose fermented colonies. You know why we are using selective media? I just told, why are we using selective media? So that other enteric microorganisms do not overgrow. So in these selective media, they contain about 1% salt. This also inhibits the growth of other enteric organisms. In mostly we use TCBS agar which has sucrose. So when this sucrose is fermented, you will be able to see large yellow colonies. This is hemodigestion seen on blood agar plate caused by Vibrio cholerae. This is TCBS agar plate. This is the yellow colonies that you can observe. This is string test. Okay. What is string test? You have taken a colony from these plates and added sodium deoxycholate to this. So when you do this, this material, it loses its turbidity and it becomes sticky like this. So this, when you pull this loop from the colony, you will see a string is formed. This is positive string test that you can observe in Vibrio cholerae. When you perform the culture smear, you will be able to see short curved gram-negative bacilli again, and when you perform hanging drop method, you will again see darting motility. Okay, laboratory diagnosis for the biochemical reactions. It is catalase positive and oxidase positive. Indole will be positive. Citrate may be utilized or not, it's variable. In TSI broth, you will be C acid by acid, A by A reaction with no gas, no H2S. Okay. String test positive, I showed you hemodigestion on the bladder guard. Other methods, automated systems such as Maltitov and Vitek you can use. Or you can perform antigen detection, which is used in the emergency cases. It's ICT test that you can perform bedside, and molecular methods such as PCR, and then you perform based on all these factors. When the organism is identified, you'll go for AST. What is the mainstay of treatment here? Mainstay of treatment is always fluid replacement. Okay, even before the antibiotics, you have to start on the fluid replacement that the patient has lost. In case of mild to moderate fluid loss, you will give ORS. In severe cases, you will use Ringer's lactate, and then when the consciousness arrives, you can replace it with ORS. Then you can add antibiotics. What antibiotics you can use here? Drug of choice remains macrolides, even in children and pregnancy. There are certain alternatives that you can use, that is doxycycline or ciprofloxacin. General measures: safe water, sanitation, proper hygiene, identifying the carriers, treating them. For chemoprophylaxis, we can give tetracycline for household contacts, but it only adds up to the antimicrobial resistance, so this is not recommended. Okay, there are certain vaccines that are available for cholera. The whole cell vaccine is composed of killed whole cell of Vibrio cholerae O1 and O139, so it covers both the serogroups. Formulations that are available are Shankol from India and Uvicol in South Korea. 2 doses are given orally, protects about for 3 years. Then there is this another recombinant B unit subunit vaccine by the name of Dukoral. Here also you have to give 2 doses, protects about for 2 years. Oral live attenuated vaccine, commercially available as Vaxchora, given as the single dose. It is recommended for the adults traveling to area with the active cholera transmission, and it gives about 90% protection at 10 days after vaccination for about only 6 months. What is 90% protection? White t-shirt in the 3rd row. The person who is drinking water. What is 90% protection? Okay, coming to halophilic vibrios. Anybody can name halophilic vibrios? We saw it in the beginning. Parahemolyticus, alginolyticus and Vulnificus. What was the speciality? They were salt loving, so they could withstand more than 60% of salt concentration. They are widespread in which environment? Obviously marine environment, because it's salty. Have you tasted ocean water? We even saw the transport medium was autoclaved sea water, even we can use for cholera, right? So these cases, they tend to occur in late summer or early rainfall when the bacterial counts are highest in the water. Clinical manifestations that are caused by these halophilic vibrios are, they can cause foodborne gastroenteritis, mostly by the consumption of seafood, and They also present as watery diarrhea or rarely as dysentery with abdominal cramps. What are the extra intestinal manifestations that you can see? Wound infection, otitis or sepsis. Remember these parts: wound infection, otitis and sepsis. We did not discuss all this in Vibrio cholerae. They are differentiated from Vibrio cholerae because they have peritrichous flagella. What did Vibrio cholerae have? They had polar flagellum and they showed vibratory motility. So here they do not show darting motility. Darting motility was seen where? In Vibrio cholera. So they have peritrichous flagella, they are also motile but they do not show darting motility. On TCBS agar, they do not ferment sucrose, so they will be seen as what? Green in color. In TCBS agar, Vibrio cholerae was seen as yellow color. Okay. This is also important. Where else have you seen swarming? They swarm on blood agar. Okay. Urease test is positive. Salt tolerance is about 8% of sodium chloride they can withstand, and you can go for identification through Malditoff and Whiterick. Here also you can use macrolides or doxycycline. In case of extraintestinal manifestation that are associated with these underlying diseases, the antibiotics are approved, such as diabetes or pre-existing liver disease, or there is iron overload state. Basically immunosuppression. If there is immunosuppression, you have to treat the patient with antibiotics. Okay. Vulnificus is also, the name of vulnificus also means wound forming. So this will give you an idea that this is capable of causing wound infection. Okay. So what are the clinical manifestations you can see? For example, a fisherman gets a cut on his leg while getting into the boat and he develops a wound infection. So here you can suspect Vibrio vulnificus. Okay. And this can also lead to sepsis. Sepsis in cases of immunosuppressed patients. This has the capability to ferment lactose, which differentiates it from all other Vibrios. Okay. Parahemolyticus was fermenting what? TCBS, sucrose. Here it is fermenting what? Lactose. Vulnificus, lactose. Parahemolyticus, sucrose. It was not fermenting sucrose. Vibrio cholerae was fermenting sucrose. Okay. Coming to alginolyticus. Alginolyticus can cause eye, ear and wound infections. And it has the ability to grow at more than 10% of salt saturation. Okay. You can treat it with tetracycline. Okay. That was about Vibrios. Aeromonas were initially classified under Vibrios, but now they are differentiated from Vibrios and they come under different family, which is Aeromonaceae. Okay. So there is this species of Aeromonas hydrophylla which causes red leg syndrome in frogs. But they are also capable of causing diseases in humans. These are the factors that help Aeromonas cause human infections, such as lipopolysaccharide is there. There are certain exotoxins, hemolysines, phospholipases, enterotoxins, capsular polysaccharides, and tissue adherence that is mediated by fimbriae. Okay, they are all fimbriated. What are the clinical manifestations you can see in Aeromonas? They can cause gastroenteritis, mucosal and wound infection, and bacteremia, sepsis, or even respiratory tract infections. So we had not dealt with respiratory tract infections in today's lecture, but Aeromonas are causing all the clinical manifestations we saw till now. Okay, whatever Vibrio, halophilic Vibrio, non-halophilic, and apart from that, respiratory tract infections are also caused by Aeromonas. Okay, they are also oxidase and catalase positive, same as Vibrio. They are susceptible to fluoroquinolones. Alternatively, we can use cotrimoxazole or cefepime.